Thank you. Thank you. It has been a long day, so I'll try to be as quick as possible. So today I'll be discussing hypertension genetics and to show you something related to the sex-dependent. And you are welcome also to come to the poster that will be presented tomorrow, the coming 2 days. And this work was led by Roy Zucker in my lab. So hypertension, what is it? It's a condition in which a persistent high blood pressure is in a way of a continuous risk. And you can see that it's not a specific number, but actually it's a combination of systolic and diastolic type of graph in which you don't want to be in the red area. So actually hypertension is really occurring in more than 1 billion people in the world. Many of them are not aware that they have it. And more importantly, it's really called the silent killer because it's associated with a lot of stroke, heart attack, vision loss, and so on and so forth, really a lot of symptoms. So it's a chronic disease with many complications. And in terms of number, about 1 in 4 males will have hypertension, and a little bit less, 1 in 5 in females. And there is also a quite substantial difference in continent, just a global view, in which the highest ratio, 37%, is in Africa and Middle East, and less so in the other part of the world. So just to say why is it so important, because lowering the blood pressure, even 10%, not much, will reduce the stroke but by 37%, the heart attacks by 25%, and overall death by quite substantially. So we ask, can genetics be used in hypertension control plan or clinical view? So to do so, you have to understand what's factor affecting hypertension, of course. And I just list a list of non-modifiable factors, such as nutrition and activity and so on. But today I'll be discussing the non-modifiable factors, including mainly genetics and sex. Of course, there is another factors that I'm not talking much about, which is age, but hypertension prevalence increases with age. So this is something that we know. The data that we are using is of Biobank, as we heard a lot about it. It's 500,000 people that were recruited between 2006 to 2010. And more importantly is that they do have a lot of information on the medical record, on lab procedure, on diagnosis, interview with nurses, and so on and so forth. To our discussion, they do have also the genetics, the genotyping, the exome, and recently also the whole genome coming up. So it's really a very rich resource. So the research goal, can we not only talk about hypertension, but make some interpretation? And in order to explain what I mean by that, I need to introduce you to PWAS, not FWAS, but PWAS. PWAS is a gene-based association method that was developed in our lab a couple of years ago. And the idea is rather simple. Instead of just going directly from the variant to the phenotype, like we are doing in GWAS, let's say, you go through the protein. So you understand what happened to the protein, and then you go to the phenotype. In a more specific, more technical aspect, what you take is all the variants from N sample to M variant. And we also included 2 heritability model, the recessive one, the dominant one, and also the hybrid one. And then we connected to the phenotype, in this case hypertension, and we end up adding a lot of covariates, about 180. I won't discuss this much. So altogether, we are only looking at variants in the coding region and splicing, and we are having on average 36 variant per gene, just on average. Okay, so what's the idea? The idea is to take a tool that we develop in the lab called FIRM, which is a machine learning based understanding what happened to the protein function directly. And then we calculate and provide the gene effect, directly score or gene effect by heritability model, the 2 or the 3 model. And then we use the information from biobank as a control and cases, and we ask whether the case and control of the gene effect of PWAS method will different, and if so, that will list the gene list that we are having statistical significant in PWAS term. I think it's that's the idea. Just to show you how a gene look like, this is a gene. You can see by color the domain, and in this lollipop, you can see the variants. And the color is just to give you a feeling of whether they destroy the protein in red or not much in green and so on. And also we have other information such as increasing the risk, decreasing the risk, and I just mark the best variant of GWAS, just to give you the feeling. Of course, it's much lower than the value of GWAS that is needed. Okay, so once we do this, we look at dominant, recessive, and hybrid model. And to do it a little bit fast, we all understand what's the GWAS, and it's very simple, it's very direct, but it has a lot of limitation. I just listed, I mean, it's very hard to make interpretation from GWAS. And the mapping of variant to gene is not trivial, of course. We discuss it throughout the day. And the statistic is pretty harsh. So those issues are treated by PWAS. We provide not only additive model, but also recessive and dominant. The interpretation is quite immediate because we are talking about protein. The mapping is basically trivial because we started with something that we can explain. And the statistics is much better because we are only talking on 18,000 genes rather than millions of variants. So just to give you a feeling, this is the method that I describe the firm, and the interpretation on protein function is that we have a 0 to 1 version of the gene effect, because 0 means that the protein is dead, not worth anything, and 1, nothing happened. And in addition, we have this difference between GWAS and PWAS that we can really deal nicely with compound heterozygosity, which GWAS cannot, at least not in a default format. So when we take everything that GWAS told us as of now, we are having about 1,400 genes related, but of course, we don't have really evidence for many of them. When we compare what the result from PWAS, we end up with 70 genes, and only partially overlap with what GWAS was telling us. So that was very interesting and maybe not expected. But the answer is that, first of all, 18% of the genes are recessive. So as I mentioned, GWAS doesn't deal that well with recessive inheritance. And this is, you can see it here. And another point is that when we look at the top list, I just list the top of these 70, you can see by the color that about half of them are protective gene, about half of them are increasing risk. So that's half and half. And that's quite interesting, and it doesn't happen in many phenotypes. So just to wrap up this part, I showed you that about 50% are protective and other are risk. I discussed 70%, 18 of them are recessive. So the last part is really, I would like to discuss about the sex dependency. Now, if we take the UK Biobank and split it now to female and male, those are the numbers. There are more females in the database. However, as I already mentioned, the male, we have 40,000 and female 33,000, 34,000. So we analyze the same way that I told you, but now we use each species, male and female, as if it's independent cohort. And when we did this, we looked at the GWAS and we found about 100 variants. Again, it's imputed variants, quite a lot of work behind it. But we can see even in the GWAS that there are few cases in which the difference between female and male is a few order of magnitude in terms of p-value. That was very interesting, and we wanted to look, can PWAS find those differences? And this is what you see here is each blue is male and pink is female, of course. And you can see that if you look at the hybrid, the distribution of male and female are not the same. And if you just look at male versus female, you can see that we have many more female genes that were not discovered by male. Only one gene was discovered by male, which is quite unique in terms of many other phenotypes that we looked at. So again, one gene is shared. There is no exclusive male gene. There are 4 exclusive female genes, and the rest are shared. So here is just a focus on the 22 genes that were female specific. And if you look at the top 10, this one in dark blue, dark color, is the one that is shared between male and female. It's a well-known gene for autoimmunity. So we look at the top 10 just to make it simple for presentation. And to our surprise, almost all of them in yellow are immune-related genes. Actually, they are expressed in tissues that are very related to immune stories, such as spleen, bone marrow, and so on. So just to talk about the top one, this is an immune related set of genes. Actually, it's in a location that is very important for regulating immunological response. This is the set of genes, and this is the phylogenetic relation. This is the Li6, very famous one group. And this guy is sitting directly inside. And we can also show by validation that an intron genes that we didn't look at, as you can imagine, was associated with hypertension in East Asian populations. So we have quite a lot of validation for this. The last point is the PRS that we discussed quite a lot. The PRS is really the simple way to say how well the person is situated in a population in terms of its genetics. And more importantly, I'll skip this for the time. More importantly, although I'm okay on time, more importantly, if you just look at, let's say, age 65, and you ask yourself, the 10% that are the lowest PRS, namely they have the best genetics, they do have a 10% a probability of prevalence of hypertension. If you go to the top one, which is 99% or 90%, they already have 40% of chance of having, not chance, but actually diagnosed the prevalence. So we are talking about quite a strong signal in this PRS. So can we really find it? So what we did is, and by this I kind of wrap up, what I show you is that first, the female have only 22% of prevalence. This is the biobank. The male have already 32% in the biobank. And when we take one gene, this is just example gene of a protective gene, one of the half that are protective, we are able to show that if you take the 7% of the population of female, you have thousands of female that have a 1.5% less hypertension than the average. So you can see that it's already can be useful in the clinic. If you know this gene, you already have a slightly, but a lot of people. If you look at the other side, this is for male. I'm talking about few, not many, only the top percentage that have 54% of prevalence versus the 32 average. So what I wanted to show you that this gene-based PRS can be used quite easily in the population studies. And this is just one example. I didn't want to elaborate on all the other 18,000 genes, but it's there. So to conclude, what I showed you is that hypertension, which is diagnosed as ICD-10, I10, is associated with 70 significant genes. I didn't talk about if you lose a little bit the threshold, you have 127 genes. So those are candidate, but those are maybe more secure. The PWAS provide functional interpretation to target genes. PWAS also highlight genetic effect in female, mostly in female, and female-specific genes were quite enriched in immunity function. PWAS provide information on clinical utility, as I showed you with the PRS. And the hypothesis maybe is that the mechanism for hypertension predisposition may be different in female and male. I mean, it's a bit wild, but I would like to pose it here. And the status of the immune system maybe determine the tendency for this diagnosis in female. And I think I'll stop here. And thank you very much. Yeah, and you can come to the poster to talk more about it if you have any question.
Even if you are all late, I think we have time for questions because the talk.
Yeah, I did it fast. Yeah.
Thanks, Michal. Great talk. I was wondering from the methodological point of view, what you did is separating female from male. However, you can also test for the specific interaction of the SNP with the sex covariate, like the interaction term.
Yeah, okay, that's a good point. Actually, we did, because we are doing the same covariates, including sex and age and, any center that were taking the UK Biobank and the PC, and we just flooded with a lot of covariants in order to reduce the bias that could be directly from, and we did it with and without this age issue and without and with but this time we perform it as if there is no connection between the two, which is usually you don't do it. You just combine it in order to gain power, of course. But I must say that because the high prevalence, you can do it. I mean, the number you see, we are talking about 77,000 people with hypertension. I should maybe mention that there is another subtlety that I didn't mention here, that you can also look at the systolic view, not as a diagnosis, but just as a record. And when we repeated this for male and female, we were able to get almost the same set of genes, which was kind of nice, that it's not really so much dependent on the doctor that say, oh, you have it or not. It was more like a lab result that were good enough to predict those genes, that many of them, by the way, were not part of the known hypertension story, which is, yeah.
All right, thanks.
Thank you.
Great talk, thank you.
Thank you.
I wonder, like at the end, you kind of speculate there could be a different mechanism between men and women,
Yeah.
but maybe there is something confounding. So for instance, maybe males drink more alcohol and have different diets, and this interacts with the genetic factors or overwhelms the genetic signal in men.
Absolutely. It's a very good point. I mean, we didn't talk at all about the interaction, but this is why I mentioned very briefly the modifiable factors, the environmental. There is a lot of them, and actually they account for quite a lot of the story. And male and female are different, and we are even trying now to see whether, for example, postmenopause and premenopause and so on, to see something on this line. The UK Biobank is not great for this because there is no really time points so well, but we can do it with other resources, and it's just, I could tell you that another 20 or 30 phenotype that we checked, we never saw it. So it's something within, it's not like the general rule, it's the exception.
Thank you.
Sure.
There's time for another question if you want. Yes.
Yeah, shall I take it up? I was going to ask a similar question, and I think the previous question asker gave me maybe a hint. Because I also did a sex stratified GWAS, and the phenotype was age at onset of ischemic stroke, so one of the potential outcomes of hypertension. And it was much less powered than your data.
Of course.
But I saw a similar trend. I saw larger effect sizes in women, and whenever a SNP was seemed to have a difference, it always had not always, almost always had a larger effect in women. And you were showing the same. And I was wondering, do you have some hypothesis?
So it's a good. I mean, I was wondering. I thought might be biased numbers. And we did this in, as I said, about 20 big phenotype, and it's not there. So it's a very unique to. I can list more than one phenotype that is like this, like hypothyroidism is the same and so on, but not many of them. And so we have some kind of internal control that it's not kind of bias in this population. And we know that some, phenotype like osteoporosis is a very female hormone story. So I'm not talking about this. I'm talking about the phenotype that was apparently unrelated. I mean, it's related in the prevalence, but not in genetics,
No, exactly.
Mendelian randomization works for all.
So we saw something similar in a phenotype that's also cardiovascular. So
it's interesting. And
it's really
you are talking about GWAS analysis or something else?
Yeah, this was a GWAS of age onset of ischemic stroke. It's not published yet, but
Yeah, it's very, we can talk about it. It's very interesting.
Thank you.
Thank you so much.
Okay.